Hi, class, and welcome to Chapter 9, which will be on muscles and muscle tissue. Part A is a good review about the different types of muscle tissue. So we'll go through cardiac, skeletal, and smooth, which will be a great review from anatomy. And then parts B and C will get a little bit more into how muscles contract. This chapter goes really well with your Physio X lab, the exercise 2 that you'll be doing about muscles. So this chapter will hopefully serve as a good introduction and go kind of along well with that Physio X lab. So all about muscles and how they contract and how we can test those muscle contractions. So your muscles make up nearly half of your body's mass. Muscles can transform chemical energy, which is ATP, into mechanical energy, which just means motion, resulting in a force. To investigate the muscle, we'll look at the types of muscle tissue, the characteristics of muscle tissue, and then muscle functions. Terminologies, whenever you see the prefix myo, mis, or sarco, those are all prefixes for muscle. So for an example, the sarcoplasm is the muscle cell cytoplasm, so everything within the muscle cell. We'll go through quickly the three types of muscle tissue, and only skeletal and smooth muscle cells are elongated and referred to as muscle fibers. So this is an important thing to note. Whenever the PowerPoints or I use the word muscle fiber, that just means a muscle cell. So here's some really good information reviewing skeletal muscle that I'm going to let you guys review on your own. We talked a little bit about this in histology. There are striations, it's voluntary, it contracts rapidly, so it tires easily, and it's extremely powerful. So your skeletal muscles are the ones that you probably learned in anatomy: your biceps, your triceps, your rectus abdominis, your rectus femoris, your sartorius, your quadriceps. The key words for skeletal muscle are skeletal, striated, and voluntary. Cardiac muscle is only found in the heart, makes up the bulk of the heart walls. It's also striated, but it means it's involuntary. It cannot be controlled consciously. It contracts at a steady rate due to the heart's own pacemaker, but the nervous system can increase the rate. So keywords for cardiac muscle are cardiac, striated, and involuntary. And then smooth muscle is found in the walls of your hollow organs like your stomach, your urinary bladder, your airways, so your bronchioles. It's not striated, it's involuntarily, meaning it cannot be controlled consciously. So it's being controlled subconsciously without you knowing it. Keywords for smooth muscle are visceral because it's referring to the organs, non-striated, and involuntary. So this is just a good review. Feel free to pause the lecture to just review these three types of muscle tissue. All muscles share four main characteristics. They're excitable, meaning they can respond to stimuli. Contractility means they can shorten forcibly when stimulated. Extensibility means the ability to be stretched. And elasticity means their ability to recoil to resting length. The four important functions, they can produce movement, such as responsible for all of your locomotion and manipulation, like walking, digesting, and pumping blood. They help you maintain your body posture as you're sitting here at your computer. They help your back sit up straight, and body position. They help to stabilize your joints, like your ball and socket joints of the shoulder and hip, as well as your synovial joints, like your knee and elbow. And they also generate heat as they contract. So this is why if you're extremely cold, which doesn't happen often in Southern California, but if you are cold, you'll start to shiver, and that shivering produces heat. Skeletal muscle is its own separate organ made up of different tissues with its own nerve and blood supply, connective tissue sheaths, and attachments. Each muscle receives a nerve, artery, and vein, and contracting muscle fibers require huge amounts of oxygen and nutrients, and that also produces waste products that need to be removed quickly. So each skeletal muscle, as well as each muscle fiber, is covered in connective tissue. You have different support cells that reinforce the whole muscle, and these are the sheaths that surround the muscle tissue. From external to internal, we have the epimysium, which surrounds the entire muscle. The perimysium surrounds the fascicles, which are groups of muscle fibers, and then the endomysium surrounds each muscle fiber. And these are just different connective tissue sheaths that surround the muscle. So here we have our epimysium surrounding the entire muscle, the perimysium surrounding the fascicle, which is made up of bundles of muscle fibers, and the endomysium is between individual muscle fibers. So it's good to know this hierarchy of how muscle tissue is arranged and the connective tissue sheaths that surround each. All muscles are attached to bones in at least two places, the insertion and origin. The attachments can be direct or indirect. And you can see here kind of where the tendon will be inserted into the bone at the insertion. And here's another great structure and organizational levels of skeletal muscle that I would encourage you to pause and just kind of read through to kind of give you kind of another review from anatomy. So skeletal muscle fibers are long cylindrical cells that contain multiple nuclei. The sarcolemma refers to the muscle fiber plasma membrane. The sarcoplasm is the muscle fiber cytoplasm, which contains many glycosomes for glycogen storage, as well as myoglobin for oxygen storage. The skeletal muscle cell has modified organelles. It has myofibrils, a sarcoplasmic reticulum, and also T tubules. The myofibrils are densely packed rod-like elements. They contain striations, sarcomeres, myofilaments, and the molecular composition of those myofilaments, which will be responsible for muscle contraction. So here's a look at a diagram of a part of a muscle fiber showing the myofibrils. One myofibril extends from the cut end of the fiber, as you can see there. You can see the dark and light striations. The nucleus will always be on the periphery, and you'll see there's tons of mitochondria packed within the muscle fiber to help with ATP. Striations are stripes that are formed from repeating series of dark and light bands along the length of each myofibril. The A bands and the I bands correspond to the dark and lighter regions. And then we have different zones and lines, which correspond. The M line will be the line of protein that bisects the H zone. The Z disc is a coin-shaped sheet of proteins on the midline. Here's a look at a photomicrograph of portions of two muscle fibers. And you can see here the light and dark bands just correspond to the different striations within the nuclei on the periphery, and one muscle fiber is shown here. A sarcomere, again, is the smallest contractile unit or the functional unit of a muscle fiber. It will consist of the area between two Z discs, which will be containing an A band with half of an I band at each end. The individual sarcomere will align end to end along the myofibril, kind of like boxcars on a train. And this is a look at the microscopic anatomy of a skeletal muscle fiber. You can see here that the sarcomere extends from one Z disc to another. And the I bands and A bands include different regions of myosin and actin filaments and how they overlap. And you can see here the thick and thin filament is just another word for actin and myosin. And these are the myofilaments that will slide past each other that causes the sarcomere to shorten in a muscle contraction. So the myofilaments, again, is the orderly arrangement of actin and myosin within the sarcomere. Actin refers to thin filaments and myosin refers to thick filaments, and they're each arranged in their appropriate place in the sarcomere. So here's again a look at the sarcomere with an enlarged view of one sarcomere that's sectioned lengthwise. The M line goes right down the middle. That's what the myosin are attached to. The Z disc will span from one sarcomere end to the other sarcomere. And you can see here the cross section of a sarcomere cut through different locations. In the I band, there will be thin filaments only. The H zone will be thick filaments only. The M line is where the thick filaments are linked by accessory proteins, and the outer edge of the A band will be where the thick and thin filaments overlap. So the molecular composition of myofilaments, again, your thick filaments are composed of the protein myosin that contains 2 heavy and 4 light polypeptide chains. They're kind of wound together with a tail and a globular head. And during contraction, the heads will link the thick and thin filaments together, forming a cross bridge or a connection between the thick and thin filaments. Your myosins are offset from each other, and they'll result in a staggering array of heads at different points along the thin filaments. Your thin filaments are composed of a fibrous protein called actin, which is a polypeptide made up of kidney-shaped G-actin subunits. Tropomyosin and troponin are just regulatory proteins that are bound to actin. So here's a look at the myosin, a portion of the thick filament, which contains several myosin molecules made up of tails, and the myosin heads, which contain the actin binding sites. And that's really important because these myosin heads will contain the actin binding sites for which the actin will bind to form a cross bridge during muscle contraction. Here's a look at a thin filament or actin. And you can see here actin consists of 2 strands of actin subunits kind of twisted together in a helix, plus the 2 types of regulatory proteins, which will be extremely important. The tropomyosin looks like this little spaghetti-like structure that's nice and thin, and the troponin is the protein that will change shape to move the tropomyosin off of the myosin binding sites in the actin when calcium binds. So, what happens during muscle contraction, really simply put, calcium will come and bind to troponin. That'll change the shape of troponin and move the tropomyosin off of the myosin binding sites. When that happens, the myosin heads are able to bind to the actin and cause a muscle contraction or shortening. This takes you through other proteins that help form the structure of the myofibril. You guys can read through this, but I'm going to kind of pass over it for now. So, not completely important. We do talk a little bit, though, about Duchenne muscular dystrophy. It's the most common and serious form of muscular dystrophies. It's a muscle destroying disease that generally appears during childhood. I had a good friend in grade school that had Duchenne muscular dystrophy, and he lived to be about 25 years old, which is much longer than the average lifespan. So it's inherited as a sex-linked recessive disease. So it's almost exclusively in males. And my friend was a male. It appears between 2 and 7 years old, when a boy would become clumsy or fall more. And the disease progresses from the extremities and then upward, finally affecting the head, the chest muscles, and cardiac muscles. So my friend, he initially could walk and do great at recess, and then eventually he would start to use a cane. And by 8th grade in high school, he was in a wheelchair. With supportive care, people with DMD can live into their 30s and beyond, but that's not as common. It's caused by a defective gene for dystrophin, which is a protein that links the thin filaments to the extracellular matrix to help stabilize the sarcolemma, which is the plasma membrane of your muscle fibers. The sarcolemma of these DMD patients will tear really easily. That allows the entry of excess calcium, which will damage those contractile fibers. Inflammation will follow. The regenerative capacity is completely lost, which means that these muscle cells aren't able to repair themselves, resulting in increased apoptosis or cell death of the muscle cells and a drop in muscle mass. And this is just a look at a boy with Duchenne muscular dystrophy. Again, it's kind of characterized if you see a boy or a child between the ages of 2 and 7, just easily falling or not having a lot of coordination, you kind of want to take a look at their muscle mass. It eventually affects their chest muscles and cardiac muscles. So the weakness will eventually just affect their heart beating and the ability to breathe. But with supportive care, again, they can live in their 30s and beyond, but not always very common. So we're going to talk about the sarcoplasmic reticulum and the T tubules. Your sarcoplasmic reticulum is a network of smooth endoplasmic reticulum tubules that surround each myofibril. It runs longitudinally, and terminal cisterns form a perpendicular cross channel at the A and I band junction. The big thing that I want you to remember about the sarcoplasmic reticulum is that it stores and releases calcium. And calcium is the necessary ion for muscle contraction because it will be what binds to the actin to allow the myosin heads to bind. The T tubule is kind of like a protrusion or an invagination of the sarcolemma deep into the cell interior. And the T tubule is just what will carry the calcium down into the muscle fiber. The triad relationships, this is just the triad relationships between the sarcoplasmic reticulum and the T tubule. When an electrical impulse will pass by, so like we talked about before, your muscle fibers can be electrically stimulated, the T-tubule proteins will change shape, causing the sarcoplasmic reticulum proteins to change shape, which again will cause the release of calcium into the cytoplasm. And again, the release of calcium from the sarcoplasmic reticulum into the cytoplasm is what is important for muscle contraction. So here we see the sarcolemma Z disc to Z disc and the different bands and how they correspond to the overlapping of actin and myosin. And in blue, you see the triad, which is made up of the T tubule and the terminal cisternae of two sarcoplasmic reticulum. So again, what you're seeing here in light blue and dark blue is just the T tubule, so kind of the invaginations that goes deep into the cell fiber, and then the sarcoplasmic reticulum on either side of that. And again, this is what will carry the electrical impulse down. So the electrical impulse from an action potential, when your muscles are stimulated, that action potential will travel down the T tubule, and then that action potential causes calcium to be released from the terminal cisterns of the sarcoplasmic reticulum. And that calcium, again, is important for binding to troponin, that protein on actin, which will allow myosin heads to again bind to actin. So now we'll talk a little bit about the sliding filament model, which is just a fancy way of saying how these muscle fibers contract. And again, contraction of a muscle fiber means it's shortening, and that shortening of the muscle fiber will allow the entire muscle to pull on the bone that's attached to, and that's what causes your bones to move. The shortening occurs when tension generated by cross bridges on thin filaments exceeds forces opposing shortening, and contraction ends when the cross bridges become inactive. In a relaxed state, the thin and thick filaments will overlap only slightly, and the sliding filament model of contraction states that during contraction, the thin filaments will slide past your thick filaments, causing the actin and myosin to overlap more. Neither the thick nor the thin filaments will actually change their length. They're just overlapping or sliding past each other more to cause the muscle contraction. When the nervous system will stimulate the muscle fiber, myosin heads are allowed to bind to actin, which will form a cross bridge, which causes the sliding or contraction process to begin. The cross bridge attachments will form and break several times, and each time they will pull the thin filaments a little bit closer towards the center of the sarcomere. And this causes the shortening of the muscle fiber. So the Z disc will be pulled toward the M line, which is the middle of the sarcomere. And you can see here how the rest of the zones and bands will change. So here's a fully relaxed sarcomere of a muscle fiber. You can see here during contraction, the sarcomere shortens and the actin and myosin are sliding past each other. The decision to move is activated by the brain, and the signal will be transmitted down to the spinal cord to different motor neurons, which will then activate your muscle fibers. Neurons and muscle cells are excitable cells, and they're capable of action potentials, which means that these excitable cells are capable of changing their resting membrane potential voltage. The action potential will cross from the neuron to the muscle cell via the neurotransmitter called acetylcholine. And acetylcholine is a very important neurotransmitter that is important in muscle contraction. Ion channels are important. They play the major role in changing the membrane potential. And there's two classes of ion channels. They can be chemically gated ion channels, which are opened by chemical messengers such as neurotransmitters. So, for example, acetylcholine receptors on muscle cells, or voltage gated ion channels, which will open or close in response to voltage changes in membrane potential. So here's just a look at a chemically gated ion channel, which will be bound to a chemical messenger like acetylcholine that's shown with these green circles, or a voltage gated ion channel, which will allow the passage of ions through. The anatomy of motor neurons and the neuromuscular junction is important to review. Skeletal muscles are stimulated by somatic motor neurons, and axons will travel from the central nervous system to skeletal muscles, and it will divide into many branches as it enters the muscle. The axon branches will end on the muscle fiber, forming what we call the neuromuscular junction, or the junction between the neuron and the muscle, also known as the motor end plate. And each muscle fiber has one neuromuscular junction with one motor neuron. So here we see here kind of the pathway that these impulses travel down through the axon of the motor neuron into each muscle fiber. And again, the neuromuscular junction is just the region where the motor neuron contacts the skeletal muscle. It will consist of multiple axon terminals from that nerve and the underlying junctional folds of the sarcolemma. So the axon terminal and the muscle fiber are separated by a gel-like space called the synaptic cleft, and stored within the axon terminals, or the end of that nerve axon, will be membrane-bound synaptic vesicles that contain that neurotransmitter acetylcholine. We have infoldings of the sarcolemma called junctional folds, and they will contain millions of acetylcholine receptors. And the neuromuscular junction, NMJ, consists of these axon terminals, the synaptic cleft, and those junctional folds. And this is a great picture here showing the axon terminal of the motor neuron, the synaptic vesicles containing the acetylcholine, which will eventually be exocytosed out of the axon terminal. The acetylcholine will travel through the synaptic cleft, bind to receptors on the junctional folds of the sarcolemma. And when acetylcholine binds to those receptors, that is what will pass or begin the action potential that will travel into the muscle fiber. The big picture of skeletal muscle contraction is looking at the events of the neuromuscular junction, the muscle fiber excitation, so the action potential traveling through the muscle fiber, then what we call excitation contraction coupling, which will be kind of the combination of exciting the muscle fiber and the actual contraction, and then the cross bridge cycling. And again, the cross bridge means the connections that are formed between those actin and myosin filaments. And the cycling just means that it goes over and over again. These four main events are shown in this diagram. And I would encourage you guys, questions that I ask you from chapter 9 will be really associated with these four main events of skeletal muscle contraction. So pause the slide, read through these, and then we'll go through them one at a time. So events at the neuromuscular junction, the action potential arrives at the axon terminal, calcium channels open, calcium enters the motor neuron, calcium causes the release of the acetylcholine neurotransmitter into the synaptic cleft, which is the space between the neuron and the muscle fiber. Acetylcholine will diffuse across to its separate receptors, which are sodium chemical gates on the sarcolemma. When acetylcholine binds to these receptors, it opens those gates, allowing sodium to enter, resulting in what we call end plate potential. Acetylcholine esterase is an important enzyme that degrades or gets rid of excess acetylcholine. And this is really important. If you have too much acetylcholine traveling into your muscle fiber, you're going to be causing too much excitation of that muscle fiber. So we want this enzyme, acetylcholine esterase, to degrade or get rid of excess acetylcholine so that your muscle fibers aren't contracting forever. So here's step one. Step two, opening the calcium channels. Step three, acetylcholine is released. It diffuses across the synaptic cleft, binds to receptors. And then step five, this binding opens what we call chemically gating ion channels that causes kind of the passage of ions to either enter or exit the muscle fiber. This produces a local change in the membrane potential, which we called end plate potential. Step six, acetylcholine effects are terminated by its breakdown by acetylcholine esterase. Here's a great animation that explains exactly what we talked about. So you could probably watch this animation and feel a lot more comfortable with that. Many toxins and drugs and diseases will interfere with events at this neuromuscular junction. So, for example, myasthenia gravis is a disease characterized by drooping upper eyelids, difficulty swallowing and talking, and generalized muscle weakness. And it involves shortage of acetylcholine receptors because the person's acetylcholine receptors are attacked by its own antibodies. So this suggests that it's an autoimmune disease. So if you don't have acetylcholine receptors, those muscle fibers will never get stimulated. So how do we generate an action potential, which means basically how do we excite this muscle fiber when acetylcholine binds to receptors? Well, the resting sarcolemma or the plasma membrane of that muscle fiber is polarized, meaning there's a voltage that exists across the membrane. Inside of the cell is negative compared to outside of the cell, and the action potential is caused by changes in electrical charges. This occurs in three steps. We generate an end plate potential, then we depolarize the cell, which means we send out an electrical signal through it, and then repolarization means we get the muscle fiber cell back to normal. End plate potential is when the acetylcholine binds to receptors, causing chemically gated ion channels to open. Sodium diffuses into the muscle cell. Some potassium moves outward, but because sodium diffuses in, this makes the inside of the cell more positive. And this results in what we call depolarization or an end plate potential. And that is shown here by these three steps, which you can read through on your own. Depolarization just means a generation and propagation of that action potential. So the large influx of sodium triggers an action potential that's unstoppable and will lead to muscle fiber contraction. The action potential travels across the sarcolemma from one voltage gated sodium channel to the next, causing that area to depolarize. And you can see this action potential area traveling along the sarcolemma. Repolarization just means restoring back to normal. So the sodium channels will close, potassium channels open, potassium effluxes out of the cell, which just means it brings everything back to normal. The refractory period refers to the muscle fiber that cannot be stimulated for a specific amount of time until repolarization is complete, and ionic conditions of the resting state will be restored by the sodium-potassium pump. Here's a look at the recording of an action potential in a muscle fiber. And this just shows how normally the membrane potential, the inside of that muscle fiber cell is about -90. When acetylcholine binds to those receptors on the muscle fiber, that causes sodium channels to open. Sodium rushes into the cell, causing the inside of the cell to become more positive. And that's why we get to about positive 30 here. At that point, we get our action potential peak, which just means we've excited the cell. The sodium channels will then close, potassium channels open, and potassium will exit the cell. And when this positively charged potassium exits the cell, the inside of the cell will become more negative again, which brings everything back to normal or repolarization. Excitation, contraction, coupling are all the events that transmit the action potential along the sarcolemma and that are coupled to the sliding of the myofilaments. So we call it excitation, contraction. The action potential will be propagated along the sarcolemma and down into the T tubules, where voltage sensitive proteins in your tubules will stimulate calcium release from the sarcoplasmic reticulum. The sarcoplasmic reticulum will release calcium, which will be important for muscle contraction. The action potential is extremely brief and it will end actually before the contraction is seen. So here's a look. Excitation, contraction, coupling, again, is the sequence of events by which the transmission of an action potential along the sarcolemma will lead to the sliding of the myofilaments. Step one is that the action potential propagates along the sarcolemma down the T tubule, releasing calcium ions from the sarcoplasmic reticulum. The calcium will then bind to the protein troponin, which again is that protein on the actin. And when calcium binds to troponin, it removes the blocking action of those spaghetti-like proteins called tropomyosin. Contraction will then begin because the myosin heads can attach themselves to the actin in what we call a myosin cross bridge. Please look at this animation. This is a good chapter, especially good chapter to look at the animations. At low intracellular calcium concentrations, tropomyosin will always block the active sites on actin, so the myosin heads cannot attach to actin and the muscle fiber will remain relaxed. Voltage sensitive proteins in the T tubules will change shape, causing the sarcoplasmic reticulum to release calcium. When calcium concentrations are high, calcium will bind to troponin. Troponin will change shape and move those tropomyosin proteins away from myosin binding sites. Myosin heads are then allowed to bind to the actin, forming this cross bridge, and this cycling process is initiated, which causes the sarcomeres to shorten and the muscle to contract. When nervous stimulation stops, calcium or excess calcium will be pumped back into the sarcoplasmic reticulum so that contraction ends. Here are the 4 steps of the cross bridge cycle. Cross bridge formation, working, or the power stroke, where the myosin head will pivot and pull the thin filament to slide past it towards the M line. Cross bridge detachment, which requires ATP to attach to the myosin head, causing the cross bridge to detach. And then the cocking of the myosin head, which just means that we get energy from the hydrolysis of ATP, which gets the myosin head back into a high energy state, ready to pull the actin again. This energy will be used for the power stroke in the next cross bridge cycle. So here's the cross bridge cycle about how the cross bridge forms, the power or working stroke. The cross bridge detaches, which always requires ATP, and then the cocking of the myosin head to get it back into position. Another great animation on that. Rigor mortis occurs 3 to 4 hours after death when the muscles begin to stiffen. So 3 to 4 hours after death, the body muscles begin to get extremely stiff. And this is because intracellular calcium levels increase because ATP is no longer being synthesized. So calcium cannot be pumped back into the sarcoplasmic reticulum, which results in the cross bridge formation, meaning that the myosin will be constantly attached to actin, causing like this constant contraction or extremely stiffening of the muscles. It also results because ATP is also needed for the cross bridges to attach from each other. and ATP energy isn't being produced when you're dead. So your muscles will stay contracted until your muscle proteins break down, causing the myosin to release. And that is the end of part A. Part A was probably kind of the bulk of this chapter where I'll get the majority of your questions from. Parts B and C, I'm just going to go over relatively quickly. And that's because parts B and C of this chapter are really going to be what your lab focuses on. So you guys are responsible for the information in parts B and C of chapter 9, but you guys will get a lot of this information by working through those seven lab activities in that PhysioX lab. So please work through those lab activities because that will directly relate to the information you need to know in chapter 9. Part B goes through what's the difference between muscle tension, whether it's an isometric contraction or isotonic contraction. That means that a contraction may or may not shorten the muscle. So an isometric contraction means no shortening exists, and an isotonic contraction meaning the muscle does shorten because the muscle tension exceeds the load. The force and duration of a contraction varies. Each muscle is served by at least one motor nerve that can contain axons up to hundreds of motor neurons. And like we talked about previously, the axons will branch into terminals, each of which will form a neuromuscular junction with a single muscle fiber. A motor unit, is the nerve muscle functional unit. And the motor unit consists of the motor neuron and all the muscle fibers that it supplies. So the smaller the fiber number, the greater the fine control. So for example, muscle fibers from a motor unit are spread throughout the whole muscle so that stimulation of a single motor unit causes only weak contraction of entire muscle. So here's a look at a motor unit consisting of one motor neuron and all the muscle fibers it innervates. A muscle twitch is the simplest contraction resulting from a muscle fiber's response to a single action potential from a motor neuron. The muscle fiber will contract quickly and then relax, as will be shown in your lab. The twitch can be observed and recorded as a myogram that you'll see in lab, and a tracing is the line recording contraction activity. The three phases of a muscle twitch include the latent period, where no muscle tension is seen. These are the events of the excitation contraction coupling. The period of contraction, which includes the cross bridge formation where the tension increases, and the period of relaxation where calcium gets pumped back into your sarcoplasmic reticulum, the tension declines to zero. Muscles always contract faster than they relax. So here's a look at the muscle twitch and the three periods, the latent period, the period of contraction, and relaxation. There's differences in strength and duration of twitches. For example, your eye muscles contract rapidly and brief, whereas your larger calf muscles contract more slowly and hold it longer. So here are different muscle twitch kind of lengths in milliseconds, whether it's your soleus, gastrocnemius, or other muscle fibers. The graded muscle responses is just talking about the relatively smooth and strength varying that each muscle needs. And these responses are graded by the changing of frequency of the stimulation and changing the strength of the stimulation. And I'm going to kind of skip over this because you guys will be stimulating muscles in the lab simulation to generate tension, twitches, and see how different stimulations can respond to that. Wave summations result if two stimuli are received by a muscle in rapid succession, as shown by here, kind of a temporal stimulation. Muscle responses to changes in stimulus frequency can be fused or unfused, and you'll see what that means in your lab as well. So here's a temporal summation caused unfused. This is called unfused tetanus. meaning several stimuli. So your muscles being stimulated over and over again, but unfused tetanus, meaning there's a little kind of relaxation between stimuli. Muscles can be stimulated constantly into what we call fused or complete tetanus, and this can cause prolonged muscle contraction, leading to muscle fatigue. And this is what fused tetanus looks like. So your stimulations are constantly stimulating the muscle, which allow to a constant kind of fused state, which we call tetany. Recruitment refers to a stimulus being sent to more muscle fibers, leading to more precise control. So, recruitment is important for gaining more control of the muscle. So, this occurs if you want to use all of the muscle fibers. So, here we have a proportion of motor units excited as each stimulus is achieved, eventually getting to a maximum stimulus. Muscle response to changes in stimulus strength. Recruitment works on a size principle, meaning the motor units with the smallest muscle fibers are recruited first, and motor units with larger fibers are recruited as the stimulus intensity increases. Largest motor units are activated only for the most powerful contractions, and motor units in muscles usually contract asynchronously, meaning that some fibers contract while others rest, and this helps to prevent fatigue. So here's a look at the size principle of recruitment. Muscle tone is constant, slightly contracted state of all muscles, and this is due to spinal reflexes where groups of motor units are alternatively activated in response to the input from stretch receptors in muscles. This keeps your muscles firm, healthy, and ready to respond. An isotonic contraction is where the muscle changes in length and moves a load. And an isotonic contraction can be either concentric, where the muscle shortens and does work, such as your biceps contracting and picking up a book, or eccentric, for example, of laying a book down, causing your biceps to lengthen while still generating a force. So here's just a look at an isotonic contraction being concentric and isometric contraction. And you guys will go over isotonic and isometric contractions in your lab as well. So I'm going to kind of skip over those. You are responsible for this information, but you'll look over a lot of it in lab. The energy for contraction and ATP, ATP supplies the energy needed for your muscle fibers to move and detach the cross bridges between actin and myosin, to pump calcium back into the sarcoplasmic reticulum after the contraction has completed, and to also pump sodium out and potassium back into the cell to reestablish that resting membrane potential. The available stores of ATP are depleted in 4 to 6 seconds, and ATP is the only source of energy for contractile activities. Therefore, it always must be regenerated quickly. ATP is regenerated by these 3 mechanisms: direct phosphorylation of ADP by what we call creatine phosphate, the anaerobic pathway, which uses glycolysis and lactic acid formation, and then the aerobic pathway. I'm going to let you guys read through these on your own as a brief review. I won't ask you guys details about these, but you should know just briefly where the ATP comes from to generate energy for contraction. And we're really going to talk about the aerobic respiration pathway when we get to more digestion, because we'll talk about especially how ATP is produced, not only for muscle contraction, but also for other areas in the body. Here's just a comparison of energy sources used during short-term exercise and prolonged duration exercise. Muscle fatigue, then, is the physiological inability to contract despite continuous stimulation. So even if you're continuously stimulating your muscles, they might not be able to contract. The possible causes could be an ionic imbalance or the increased inorganic phosphate and ATP breakdown that could interfere with calcium release. Decreased ATP and increased magnesium levels A lack of ATP is rarely a reason for fatigue, except in severely stressed muscles. Excess post-exercise oxygen consumption refers to that a muscle is able to return to its pre-exercise state only if oxygen reserves are replenished, lactic acid is converted back to pyruvic acid, glycogen stores are replaced, and ATP and creatine phosphate reserves are resynthesized. All replenishing steps require extra oxygen, so this is referred to as what we call excess post-exercise oxygen consumption, or what we refer to as oxygen debt. All right, and then part 3, and thanks, I'm glad you guys are still with me here. Part C, excuse me. And again, we're moving through parts B and C a little more quickly because this is kind of where you're going to focus your lab work. The force of muscle contractions depends on the number of cross bridges attached, which is affected by 4 different factors. The number of muscle fibers stimulated, which is what we call recruitment. So the more motor units recruited, the greater the force. The relative size of the fibers, so the bulkier the muscle, the more tension it can develop. Muscle cells can increase in size, that we call that hypertrophy, which is regular exercise. The frequency of the stimulation, so the higher the frequency, the greater the force, so the stimuli can be added together. And then the degree of the muscle stretch, meaning the muscle fibers with sarcomeres that are 80 to 120% their normal resting length will generate more stretch. So this is a look at the factors that increase force of skeletal muscle contraction, which you should know these 4 factors. Here's the length tension relationships of the sarcomeres and skeletal muscles, showing the percent of resting sarcomere length and then the tension. So the sarcomeres greatly shortened will give about 75% tension. Sarcomeres at resting length are about 100% tension. And then sarcomeres extremely stretched gives them at about 170% tension. So how fast a muscle contracts and how long it can stay contracted is influenced by the muscle fiber type, the load, and the recruitment. The muscle fiber type is classified by either the speed of contraction, slow or fast fibers, and then the metabolic pathway, whether they're using oxidative fibers for aerobic pathways or glycolytic fibers using anaerobic glycolysis. The muscle fiber type, slow oxidative fibers, that's one type of muscle fiber, or fast oxidative fibers or fast glycolytic fibers. Most muscles contain a mixture of all the fibers, and all the fibers in one motor unit are the same type. Genetics will usually dictate an individual's percentage of each type. So slow oxidative fibers are more consistent with people who use their muscles for endurance training, such as marathon, swimming. Fast oxidative fibers and fast glycolytic fibers are more important for kind of speed, quick contraction workouts. The different muscle types are better suited for different jobs. So for example, your slow oxidative fibers are important in your muscles that are for endurance activities, so maintaining posture. So the muscles in your calves and your back are slow oxidative fibers. They're usually constantly contracted. Fast oxidative fibers are important for sprinting or walking. And then fast glycolytic fibers are short-term, intense, or powerful movements like hitting a baseball. So here's a look at different types of factors influencing velocity and duration of skeletal muscle contraction. And this is a great chart to review slow and fast oxidative fibers and fast glycolytic fibers, their metabolic characteristics, the activities best suited for, and structural characteristics. Load and recruitment. You should know the difference between load and recruitment. Load refers to the muscles contracting fastest when no load is added. And the greater the load, the shorter the duration of contraction. And the greater the load, the slower the contraction. Recruitment refers to the more motor units contracting, the faster and the more prolonged contraction. So here's a look at the influence of load and duration and the velocity of muscle shortening, showing over time a heavier load will have a certain distance being shortened of that muscle fiber, whereas an intermediate load and a lighter load. Aerobic exercise then refers to activities like jogging, swimming, hiking, which leads to an increase in muscle capillaries, an increase in the number of mitochondria and myoglobin synthesis. And this just results in greater endurance, strength, and resistance to fatigue, which could convert fast glycolytic fibers into fast oxidative fibers. Resistance exercise, such as weightlifting, leads to muscle hypertrophy or increasing the size, increased mitochondria, which is important, myofilaments, glycogen stores, and connective tissues, also increasing muscle strength and size. Muscles must be active to remain healthy, and disuse atrophy, which refers to the degeneration and loss of mass, refers to it's due to the immobilization or loss of neural stimulation, and this can begin almost immediately. Muscle strength can decline at about 5% per day. Paralyzed muscles, especially, can atrophy to 1/4 the initial size. So, this is why it's extremely important to always do weight bearing exercises. Smooth muscle is found, like we talked about, in the most hollow organs. Smooth muscles have two layers of sheets we call a longitudinal or a circular layer, and their contractions in both layers either cause the muscle to shorten or to constrict, and alternating contractions and relaxations of these muscle layers will eventually squeeze substances through the lumen of hollow organs like your digestive tract. Smooth muscles are spindle shaped, one nucleus, no striations. They contain varicosities, which are bulbous swellings of nerve fibers, and varicosities just store and release neurotransmitters. So here's a look at the innervation with the varicosity of the smooth muscle. Some more differences between smooth and skeletal muscles. I'll let you guys read through those on your own, kind of how action potentials are generated. And a couple more differences between smooth and skeletal muscles that I'm going to let you guys read through on your own, because I don't ask you guys too many differences besides what's seen in this chart, the differences between skeletal, cardiac, and smooth. And these are probably two good charts to just study as well. And this one and this one. Actually, to be honest, these four charts include a lot of information of which I would test you guys directly from this chapter. The contraction of smooth muscle is more kind of a slow, synchronized contraction versus kind of the more abrupt contraction of skeletal muscle. So I'm going to let you guys read through the contraction of smooth muscle, because it's very similar to skeletal muscle, but it's more slow and just much more slow and versus the abrupt contraction of the skeletal muscle. The energy efficiency of smooth muscle is really important. It contracts slower and relaxed, but maintains the contraction for a prolonged period with little energy lost, which is very important in smooth muscle. Hormones and local chemicals help to regulate contraction of smooth muscle, which is important. There's different types of smooth muscle, unitary smooth muscle versus multi-unit smooth muscle. And that really takes us through the end of chapter 9. Thank you for listening. Focus your work on doing that lab. And I tried to highlight, especially in part A, the important aspects of this chapter in muscle contraction.